protein synthesis. So if you remember back in topic 3, we mentioned that genes are made up of or make up our DNA, and DNA is found in almost all living organisms, and it directs protein synthesis. So it tells it's a code for how to make proteins. So in this case, some examples we talked about before of proteins were enzymes like lactase. Remember, lactase is a protein that breaks down the sugars in lactose. So people that are lactose intolerant might not have lactase, and that's why they get sick when they have milk products. Hormones are also proteins, like insulin in our body. So our pancreas creates insulin to help decrease the spike in sugar or balance out sugar levels. We have what's called antibodies that are proteins in our body that help with the infection fighting and also our immune system response. We have hemoglobin, which is found in our red blood cells, which carry our oxygen throughout our body on the red blood cells. Cell membrane is also made out of proteins and also receptor molecules. So these are all different types of proteins that are made in our bodies. There are, of course, more, but just to get started, these are some. And we're going to go through the process of how they are made. So what are proteins made up of? So we already talked about how we have nitrogen bases that code our genes or our genetic information. This will lead to what are called amino acids. So proteins are made up of different amino acids that come together and form one protein. So there are about 20 different amino acids out there in our body. Different combinations of amino acids are going to give different proteins produced. So it's kind of like a puzzle almost, where you're matching different amino acids together and they're going to make one protein. So the amino acids formed are determined by the bases in our sequence of our DNA. So it's formed based on the coding of the nitrogen bases. So remember our A, T, C, G, adenine, thymine, guanine, and cytosine, those are going to code for what specific amino acid is going to be put so that it makes a protein. So one gene will make one protein. So one strand or one segment of your DNA with all of those multiple ATCGs that are paired up is going to make only one protein. So protein synthesis will then rely on 3 different types of RNA. So we have what's called rRNA, we have mRNA, and we have tRNA. And before we go into protein synthesis, we have to talk about the difference between DNA and RNA. Okay, so we already know what DNA is. We know it's located in the nucleus. It's what codes for everything in our body. And specifically in this topic, we're going to see DNA in the nucleus and in the cytoplasm. Remember, in cell division, that's when we see it outside of the nucleus, is when the cell is dividing. With DNA, it has 2 strands, so it's double-stranded, so like a helix, right? It twists around itself. And it's made up of sugar and 4 nitrogen bases. So cytosine, guanine, adenine, and thymine are our 4 nitrogen bases. Whereas on the other hand, RNA, specifically mRNA, is going to be found in the nucleus and the cytoplasm. It's going to be seen in both places. It's only 1 strand, so it's half the amount of DNA. And it still has a sugar, but the sugar is different. It's called ribose, so that's where the R comes from. And it has 4 nitrogen bases still, but one is actually different. So we still have cytosine, guanine, and adenine, but now uracil is in place of thymine. So DNA has thymine, mRNA has uracil. Okay, they don't both have the same one. All right. Here's an example. If you watch Game of Thrones, you know this is Khaleesi. Look at her ringlets, her hair. So RNA, the prof says, can you show me DNA and RNA visually? RNA is 1-stranded, DNA is 2-stranded. Okay. So talking about the nucleic acids, so our nitrogen bases. The difference again, DNA has thymine, whereas RNA has uracil. But they both have cytosine, adenine, and guanine. The only thing that's different is U instead of T in RNA. Okay, so again, DNA has 2 strands. So if one side of DNA is ATCG, remember we can find the complementary side by finding the Chargaff partner. So A is T, T would go with A, C would go with G, and G would go with C. Okay. Now, in this case, one mRNA strand is actually going to look a little different. So if we're going to copy this DNA strand on the right here, instead of a T, you're going to have a U on mRNA. Okay, so in this case, mRNA is the complementary strand of the coding strand or the given strand of DNA. So again, the difference first is the sugars. So DNA has deoxyribose, RNA has just ribose. And then if we start with the complementary of A, it would usually be T, but in mRNA, it's going to be U. Okay, and then everything else is the same type of complementary. So T would still go with A, C would still go with G, and G would still go with C. So the only thing that's changed in terms of the complementary strand is the U instead of a T in RNA. Okay, so one thing to remember, your mRNA strand is the complementary strand. So complementary strand to the coding DNA. Okay, so complementary strand to the coding DNA. But notice that on DNA, the other side is called the non-coding. Notice that they are equal, or they are the same, the mRNA and the non-coding DNA, but the only thing is that U is in place of T. Okay. So here's Howard again. If I had to choose between DNA and RNA, I'd choose RNA because it has U in it. Uracil is in it. Good. So different types of RNA that we're going to see in protein synthesis. The first one is messenger RNA or mRNA, which is the first example we did. mRNA is found in the first stage of protein synthesis. It's involved in what's called transcription. And we'll talk about what transcription is in a second. But essentially what mRNA does is it photocopies DNA and carries that DNA message from the nucleus into the cytoplasm to the ribosome. So remember in that chart comparing DNA and RNA, we said that RNA is found both in the nucleus and in the cytoplasm. So essentially it starts in the nucleus, it photocopies the DNA that we want to create, and then it comes out to the cytoplasm to make the protein. Okay. The second type of RNA is called ribosomal RNA or rRNA. This makes up the ribosomes. Remember in Science 10, one of the organelles that you had to know was ribosomes in animal cells and also plant cells. These ribosomes are what produces proteins. And so rRNA is what makes up the ribosomes, and ribosomes will have a play in protein synthesis. The last kind of RNA is tRNA or transfer RNA. And transfer RNA will carry the amino acid that is coded to the code of the mRNA. So the mRNA is the code or the sequence, and then the tRNA will bring out the amino acid to make the protein. Okay. In terms of location, mRNA is found in both cytoplasm and nucleus. rRNA and tRNA are only in the cytoplasm, so they only have roles in the cytoplasm. You don't see them in the nucleus at all. But all RNA is produced in the nucleolus, which is in the nucleus. Okay, so here we go, 3 types. So these pictures, these just will make more sense once we go through the steps of protein synthesis. Alrighty, so protein synthesis itself occurs primarily in the ribosomes, so that's where proteins are produced. Essentially, it needs DNA to have instructions for what proteins are produced and in terms of what kind of amino acids are brought out. Here, they get the message from the nucleus, from the DNA, which is cut in half by the RNA, and then that RNA carries it out to the cytoplasm towards the ribosome. And protein synthesis occurs in 2 steps. So the first being transcription, the second being translation. So transcription happens in the nucleus. That's the very first step. And essentially what's happening here is DNA is photocopied by RNA. So we produce RNA. And then that RNA is carried out into the cytoplasm and then goes through what's called translation. And then we produce a protein. Okay, so an easy way to remember which one comes first is that transcription has a C in it. So C comes before L. Translation has an L in it in the alphabet. So transcription is always before translation. Okay. Protein synthesis. So remember, step 1, transcription happens in the nucleus. Step 2 happens in the cytoplasm, and that's translation. This is just a little overview. It'll make more sense once we do the steps. Okay. All right. Step 1, transcription. So transcription again occurs in the nucleus. This is when we have mRNA photocopying DNA. So DNA is our template. Because DNA is double-stranded, it's way too big to leave the nucleus itself. So we have to photocopy half of it and make RNA, and then the RNA can leave. So if the DNA sequence is this, so AAT, TCC, GGA, this is our coding. This is our DNA coding strand. Okay, remember we mentioned that earlier. If this is our DNA coding strand, then RNA is going to be the complementary strand to this coding strand. Okay, but first, when we talk about triplets or codons, we're talking about the segments of 3 nitrogen bases together. So that each set of 3 nitrogen bases makes a DNA triplet or a codon. Okay, so if that's the DNA base sequence or a DNA coding strand, the mRNA is going to be complementary. So this is going to be the complementary strand, complementary RNA, mRNA. But remember that instead of T's, because the complementary of A is supposed to be T, but remember in mRNA or in RNA in general, we don't have T's, we're going to replace them with U's. Okay. These, again, these sets of 3 are called codons. So the sets of 3 nitrogen bases are codons. So here in each strand, we have 3 codons. Okay. Each DNA triplet has a complementary codon on the mRNA. So remember, complementary. mRNA will then exit the nucleus. So once we have this complementary mRNA strand, it has to exit the nucleus and go into the cytoplasm. So the code must then be transcribed before it becomes translated. So a good way to remember this is that transcription means you are writing out what is given to you. So in this case here, the DNA that is too big to leave, this is the template that needs to be written out. And so mRNA will write out this code in a complementary way, and then because it's small enough, it can leave the nucleus. Okay. All right, so here we have our DNA strand, just another visual example of this. We have our DNA strand. And then on the top end, we have what's called the non-coding strand. On the bottom, we have the coding strand. So this is the strand that wants to be photocopied. Okay, this is the strand that wants to be photocopied. But remember that RNA is going to be complementary. So it's not exactly the same in terms of photocopying. It's going to be the complementary pairs. But remember, wherever there's a T, it gets replaced with a U. So if we have a C, the complementary of C is going to be G. If we have an A, we want to put a T there, but because it's RNA, it's actually going to be U, and then etc. So G is going to become C, T is going to become A wants to become T, but because there's a mRNA, it's going to be U, C, G, G. Okay, so then now this is our mRNA strand. So transcription, we're using DNA as a template to build our mRNA. Good. So our mRNA codons, okay, each codon codes for one amino acid. So on your yellow data sheets, there's a mRNA codon chart, and there you're going to see the codons or the triplets of mRNA, and each of them are going to have an amino acid that is the code for it. So we have what are called start codons. So some may code for start or initiator codons. So that's going to start the beginning of the protein. We might have some that are called stop codons. So in this case, these are going to stop or tell us that this is the end of the protein. And then in that case, that's what makes up the strand of the protein and the amino acids for the one protein. So AUG, if you see AUG, that's a start codon. If it's at the beginning, only at the beginning of a strand. Okay. If it's anywhere in the middle, then it's going to be a methionine instead for an amino acid. Okay. So here, if it starts as AUG and then it continues, etc., then it's a start. Okay, but if let's say it's in the middle, so we have CCG, AUG, GGG, then it's going to be a methionine if it's in the middle. Okay, so if code AUG is in the middle, it must code for methionine. So again, all of these amino acids are found on your data book or your data sheet. So here's some practice for you. Try this out if you have the DNA strand. Now, if it doesn't tell you if it's coding or non-coding, then you always assume it's coding strand. Okay, so if it's a coding strand, that means the mRNA is going to be its complementary. Okay, so if we have ATG, GTC, TCG, the complementary would be UAC, CAG, and then AGC. In terms of how many triplets are codons, well, remember each triplet is 3 nitrogen bases together. So this is 1, 2, 3. So here we have 3. Awesome. So that's transcription. So first you got to transcribe the mRNA codon from the DNA. And then in translation, we find the amino acid. Okay, so here, so this is step 1. Here is step 2. So step 2 now, we're going to use the mRNA strand to find the amino acid, and then the amino acid chain is going to become our protein. Okay, so again, this is our mRNA codon sheet key that will tell us what each codon translates to in terms of amino acid. Okay, so reminder, nitrogen bases are classified as either purines or pyrimidines. It's also in your data sheet, so you don't have to memorize it. So uracil is just another pyrimidine, but for RNA. Okay, here's another example. You can try this out. And then I'll just fast forward for the answer for you. So when you're ready, you can press play again and see the answer. Okay, so that was just the mRNA codon. Remember, if they don't tell you which code or which strand DNA is given, you always assume it's coding. But because it says coding strand, that's going to be our mRNA complementary. And then from there, if you get your mRNA, you look on your data sheet and you find your amino acid chain. So remember, if AUG is at the beginning, it is your start codon. And then you just continue on and you match your mRNA to your amino acid. Again, Howard with his cheesy pickup lines. I wish I was adenine, then I could pair with you. Only in RNA. Okay, so now we get into translation. So the second half of protein synthesis. So think of translation as now you have this set of codes, you need to change the language of it, right? So translating is like changing from one language to another. So first you have the language of first you have the language of nitrogen base codes or codons. in your mRNA. Now you need to translate it into the language of amino acids. Okay, so here, translation occurs in the cytoplasm, so outside of the nucleus. It has to come to the ribosome. So this big eggplant kind of looking thing is your ribosome. That's what's going to produce your protein. This is where amino acids are assembled with the help of tRNA. So remember, tRNA is the transfer RNA. So that's going to bring, so it's this thing right here, tRNA, this little T. That's going to bring your amino acid to your corresponding codon or mRNA code. So tRNA brings the correct amino acid to the ribosome based on the complementary pair rule. Okay, so your tRNA is also a code or anticodon. So it's just the opposite or the complementary to your mRNA codon. Since there are 20 amino acids, there are 20 different tRNAs. Okay. So here is an example if you're given an mRNA sequence. So tRNA brings the correct amino acid to the ribosome when its anticodon, matches the mRNA codon. So here we have our mRNA, UUA, AGG, and CCU. So here, the tRNA would be the complementary of that. So AAU, UGG or UCC, and then GGA. Okay, and then the tRNA is how it matches. The tRNA is going to bring the amino acid. The amino acid matches, or it comes from the codon. So if you look on your data sheet, if you find UUA, not AAU, okay, you're not looking at the tRNA for your amino acid. You're looking at the mRNA. So UUA on your data sheet is going to be leucine. AGG on your data sheet is going to be arginine. And then CCU is going to be proline. Okay, so that's how you would find your amino acid chain, is based on the mRNA. The tRNA is just carrying. So if I draw it out like the picture, it's just carrying the amino acid to the corresponding mRNA. Okay, so again, mRNA from the nucleus moves to the ribosome in the cytoplasm. The ribosomes are made up of rRNA and other proteins. And then the ribosome acts like a scaffold, holding the mRNA in position while the protein is being built. It helps to join the amino acids together. So essentially what happens is the ribosome guides the tRNA down the line of mRNA to tell them which codons you're going to match next. So it's just guiding. So let's say it starts here. It's going to guide it down this way. So that's what the ribosome does. It's like a guide. Okay. All right. So translation step 1. So tRNA picks up the amino acid that corresponds to the mRNA codon, and then they match it. And then step number 2, the ribosomes move down the mRNA, and another tRNA brings the next amino acid. So again, still notice how this is the guide, right? So the ribosome is guiding it down. So it's already gotten this one done. Okay, then it guides it down to the next 3, and then it completes it, and then it continues on until you get a stop codon. Okay. Adjacent amino acids are held by peptide bonds. So here, your peptide bonds are what holds the amino acids together. Some people get this confused with hydrogen bonds, but remember, hydrogen bonds are what hold nitrogen bases together in DNA. Here, we're talking about peptide bonds, and they're going to hold together the amino acids. Okay, so we're looking at it right here. When mRNA codon reads stop, or when we get to the stop codon, so either UAG, UAA, or UGA, the polypeptide chain or the amino acid chain is done. So then it gets released. So now here, this is going to have a stop codon. So this is where it ends. Okay, so remember, if methionine is at the beginning, that means it's a start codon. So this right here is your amino acid chain, or you can call it a polypeptide chain. Polypeptide being many and then peptide bonds. So remember that each bond between amino acids are peptide bonds. So a polypeptide chain is the same thing as saying amino acid chain. So once this chain is done from start to stop, it gets released from the ribosome, and then it goes to what's called the Golgi complex. The Golgi complex is going to, if you remember from Science 10, package up the amino acid and create the protein, and then release it to your body. Okay. Here's a little stop sign in terms of a geneticist version. Okay. So again, like I mentioned, the Golgi complex or apparatus will package the protein, and then it will release it to the different parts of the body so that it can do its job. Okay. So again, translation will require many ribosomes, just a little picture here. So different ribosomes, translation will create lots of different lengths of amino acids depending on how many mRNAs you have. And each strand is going to make a different protein every time. Okay. So here on this page 43 of your workbook, you're going to have this giant circle, and we're just going to review protein synthesis, transcription, and translation in a diagram. Okay, so here we go. Transcription and translation. So again, here's our cell. There's a cell membrane. In our nucleus, we have our DNA. And then remember, each triplet of our DNA is a triplet or codon. So each group of 3 nitrogen bases is a triplet or codon. Remember that in the nucleus, the first step in protein synthesis is transcription, where mRNA photocopies or is produced as a complementary strand to the DNA coding strand. So this is transcription. When that happens, the mRNA then leaves and finds the ribosome in the cytoplasm. And remember, each 3 nitrogen bases, or each 3 groups of nitrogen bases in mRNA is called a codon. From here, the codons are matched with your tRNA or anticodon, and your tRNA is what brings your amino acid to the mRNA strand. Okay, so here we see the tRNA bringing our amino acid chain, or producing our amino acid chain, by pairing up with its complementary codon strand on the mRNA. Once the tRNA drops off the amino acid, then it leaves and then continues to bring more amino acids. Remember, between each amino acid, there's a bond called a peptide bond. And then once your amino acid chain is produced, you have what's called an amino acid chain or a polypeptide chain. And that then gets sent to your Golgi apparatus to be packaged up as a protein. And that's the end of translation, which is the second part of protein synthesis. There you go. That's it. That's protein synthesis in one diagram. So here you should know how to draw this entire thing, or you should be able to label it. Okay, there will be lots of practice in your workbook with it, and then you'll see this on quizzes and unit exams as well. All right, that's it. That's translation and transcription. Thank you for watching.